During the half-century that has elapsed since the enunciation of the cell theory by Schleiden and Schwann in 1838–39, it has become ever more clearly apparent that the key to all ultimate biological problems must, in the last analysis, be sought in the cell. It was the cell theory that first brought the structure of plants and animals under one point of view, by revealing their common plan of organization. It was through the cell theory that Kalliker, Remak, Gnarly, and Hofmeister opened the way to an understanding of the nature of embryological development and the law of genetic continuity lying at the basis of inheritance. It was the cell theory again which, in the hands of Goodseer, Virchow, and Max Schulze, inaugurated a new era in the history of physiology and pathology, by showing that all the various functions of the body in health and in disease are but the outward expression of cell activities. And at a still later day it was through the cell theory that Hertwig, Fall, van Beneden, and Strasburger solved the long-standing riddle of the fertilization of the egg and the mechanism of hereditary transmission. No other biological generalization, save only the theory of organic evolution, has brought so many apparently diverse phenomena under a common point of view or has accomplished more for the unification of knowledge. The cell theory must therefore be placed beside the evolution theory as one of the foundation stones of modern biology. And yet the historian of latter-day biology cannot fail to be struck with the fact that these two great generalizations, nearly related as they are, have been developed along widely different lines of research, and have only within a very recent period met upon a common ground. The theory of evolution originally grew out of the study of natural history, and it took definite shape long before the ultimate structure of living bodies was in any degree comprehended. The evolutionists of the Lamarckian period gave little heed to the finer details of internal organization. They were concerned mainly with the more. 1 Cellular Pathology p. 12, 1858. Obvious characters of plants and animals, their forms, colors, habits, distribution, their anatomy and embryonic development, and with the systems of classification based upon such characters. And long afterward it was, in the main, the study of like characters with reference to their historical origin that led Darwin to his splendid triumphs. The study of microscopical anatomy on which the cell theory was based lay in a different field. It was begun and long carried forward with no thought of its bearing on the origin of living forms, and even at the present day the fundamental problems of organization, with which the cell theory deals, are far less accessible to historical inquiry than those suggested by the more obvious external characters of plants and animals. Only within a few years, indeed, has the ground been cleared for that close alliance between students of organic evolution and students of the cell, which forms so striking a feature of latter-day biology and is exerting so great an influence on the direction of research. It has, therefore, only recently become possible adequately to formulate the great problems of development and heredity in the terms of cellular biology. Indeed, we can as yet do little more than so formulate them. Yet the fact that these two great lines of research, both concerned with the deeper problems of life, yet having their beginnings so far apart, have at length converged to a meeting point, is one of the more striking evidences of progress that modern biology has to show, and it sufficiently justifies an attempt to treat the cell from the standpoint of the general student of development. Let us at the outset briefly outline the cell theory as thus regarded, and indicate the manner of its historical connection with the general problems of evolution. 1. Schleiden and Schwann are universally and justly recognized as the founders of the cell theory. But like every other great generalization, the theory was based on a long series of earlier investigations, beginning with the memorable microscopical researches of Leeuwenhoek, Malpai, Hooke, and Grew in the second half of the 17th century. Wolff, in the Theoria Generationis, clearly recognized the spheres and vesicles composing the embryonic parts both of animals and of plants, though without grasping their real nature or mode of origin, and his conclusions were developed by the botanist Merbel at the beginning of the present century. Nearly at the same time, Oken foreshadowed the cell theory in the form that it assumed with Schleiden and Schwann, but his conception of Erschleim and Blaschen can hardly be regarded as more than a lucky guess. A still closer approximation to the truth is found in the works of Turpin, Mayen, Raspel, and Dutrochet, but these, like others of the same period, only paved the way for the real founders of the cell theory. Among other immediate predecessors or contemporaries of Schleiden and Schwann should be especially mentioned Robert Brown, Dujardin, Johannes Müller, Perkinge, Hugo von Moll, Valentin, Unger, Gnarly, and Henel. The significance of Schleiden's and especially of Schouwen's work lies in the thorough and comprehensive way in which the problem was studied, the philosophically breadth with which the conclusions were developed, and the far-reaching influence which they exercised upon subsequent research. In this respect, it is hardly too much to compare the mikroskopische Untersuchungen with the origin of species. During the past 30 years, the theory of organic descent has been shown by an overwhelming mass of evidence to be the only tenable conception of the origin of diverse living forms, however we may conceive the causes of the process. While the study of general zoology and botany has systematically set forth the results, and in a measure the method of organic evolution, the study of microscopical eggs. Fig. 1. A portion of the epidermis of a larval salamander as seen in slightly oblique horizontal section, enlarged 550 diameters. Most of the cells are polygonal in form, contain large nuclei, and are connected by delicate protoplasmic bridges. Above X is a branched, dark pigment cell that has crept up from the deeper layers and lies between the epidermal cells. 3 of the latter are undergoing division, the earliest stage at A, a later stage at B showing the chromosomes, and a final stage showing fission of the cell body to the right. Anatomy has shown us the nature of the material on which it has operated, demonstrating that the obvious characters of plants and animals are but varying expressions of a subtle interior organization common to all. In its broader outlines, the nature of this organization is now accurately determined, and the cell theory, by which it is formulated, is therefore no longer of an inferential or hypothetical character, but a generalized statement of observed fact which may be outlined as follows. In all the higher forms of life, whether plants or animals, the body may be resolved into a vast host of minute structural units known as cells, out of which, directly or indirectly, every part is built. The substance of the skin, of the brain, of the blood, of the bones or muscles or any other tissue, is not homogeneous as it appears to the unaided eye, but is shown by the microscope to be an aggregate composed of innumerable minute bodies, as if it were a Fig. 2. General view of cells in the growing root tip of the onion from a longitudinal section enlarged 800 diameters. A. Non-dividing cells with chromatin network and deeply stained nucleoli. B. Nuclei preparing for division. C. Dividing cells showing mitotic figures. E. Pair of daughter cells shortly after division. Colony or congeries of organisms more elementary than itself. The name cells given to these bodies by the early botanists and ultimately adopted by nearly all students of microscopical anatomy was not happily chosen, for modern studies have shown that although the cell may assume the form of a hollow chamber, as the name indicates, this is not one of its characteristic or even usual features. Essentially, the cell is a minute mass of protoplasm, a substance long since identified by Kohn, Leidig, Max Schulze, and Day Berry as the essential active basis of the organism, afterward happily characterized by Huxley as the physical basis of life. And at the present time universally recognized as the immediate substratum of all vital activity, one endlessly diversified in the details of their form and structure. These protoplasmic masses nevertheless possess a characteristic type of organization common to them all. Hence, in a certain sense, they may be regarded as elementary organic units out of which the body is compounded. This composite structure is, however, character. Fig. 3. Amsbaugh Proteus, an animal consisting of a single naked cell, x 280. N. The nucleus, WV water vacuoles, CV contractile vacuole, FV food vacuole. Istic of only the higher forms of life. Among the lowest forms at the base of the series are an immense number of microscopic plants and animals, familiar examples of which are the bacteria, diatoms, rhizopods, and infusoria, in which the entire body consists of a single cell, of the same general type as those which in the higher multicellular forms are associated to form one organic whole. Structurally, therefore, the multicellular body is in a certain sense comparable with a colony or aggregation of the lower one-celled forms. This The word protoplasm is due to Perkinge, who applied it to the formative substance of the animal embryo and compared it with the granular material of vegetable cambium. It was afterward independently used by H. von Mohl to designate the contents of the plant cell. The full physiological significance of protoplasm, its identity with the SAR code of the unicellular forms, and its essential similarity in plants and animals, was first clearly placed in evidence through the classical works of Max Schultze and Day Berry, beside which should be placed the earlier works of Dujardin, Unger, Gnarly, and Mohl, and that of Cohn, Huxley, Virchow, Leydig, Brack, Kane, and Beale. Too, this comparison must be taken with some reservation, as will appear beyond. Comparison is not less suggestive to the physiologist than to the morphologist. In the one-celled forms, all of the vital functions are performed by a single cell. In the multicellular forms, on the other hand, these functions are not equally performed by all the cells, but are in varying degree distributed among them, the cells thus falling into physiological groups or tissues, each of which is especially devoted to the performance of a specific function. Thus arises the physiological division of labor through which alone the highest development of vital activity becomes possible, and thus the cell becomes a unit not merely of structure but also of function. Each bodily function, and even the life of the organism as a whole, may thus in one sense be regarded as a resultant arising through the integration of a vast number of cell activities, and it cannot be adequately investigated without the study of the individual cell activities that lie at its root. 1. The foregoing conceptions, founded by Schwann and skillfully developed by Kalliker, Sibbald, Virchow, and Haeckel, gave an impulse to anatomical and physiological investigation the force of which could hardly be overestimated. Yet they did not for many years measurably affect the more speculative side of biological inquiry. The Origin of Species, published in 1859, scarcely mentions it, nor, with the important exception of the theory of pangenesis, did Darwin attempt at any later period to bring it into any very definite relation to his views. The initial impulse to the investigations that brought the cell theory into definite contact with the evolution theory was given nearly 20 years after The Origin of Species, by researches on the early history of the germ cells and the fertilization of the ovum. Begun in 1873-74 by Orbach, Fall, and Beachley, and eagerly followed up by Oscar Hertwig, Van Beneden, Strasburger, and a host of later workers, these investigations raised wholly new questions regarding the mechanism of development and the role of the cell in hereditary transmission. Through them, it became for the first time clearly apparent that the general problems of embryology, heredity, and evolution are indissolubly bound up with those of cell structure, and can only be fully apprehended in the light of cytological research.